Welcome to another episode from the Veterinary Medicine Digest. Today, we're diving deep into a topic that touches every corner of veterinary medicine, and indeed, every corner of our interconnected world. We're talking about a microscopic organism with a colossal impact, Escherichia coli. You've likely heard of it, perhaps in the context of food safety or a nasty bout of diarrhea. But this bacterium is far more than just a gut bug. It's a versatile, adaptable survivor, and in the age of antibiotics, its ability to survive is becoming one of the most significant challenges to global health. E. coli is a common, often harmless resident in the intestines of humans and animals. However, it's also a canary in the coal mine for a much larger problem, antimicrobial resistance. Because it's so widespread and so adept at swapping genes, scientists use it as a key indicator organism, a benchmark to measure the rising tide of resistance in our environment. Today, we're going to explore the vast and intricate world of E. coli resistance. We'll journey from the sprawling cattle farms of Turkey and the United States to the veterinary clinics of Japan, from the wild forests of Poland to the pig farms of Thailand. We'll uncover the ingenious molecular tricks this bacterium uses to outsmart our most powerful medicines, and we'll see how its fight for survival in animals has profound implications for human health. This is a story that truly embodies the One Health concept, the undeniable truth that the health of people, animals, and our shared environment are inextricably linked. So, grab your headphones, and let's begin our investigation into the secret life of a resistant superbug. Segment 1. The ubiquitous bug and its battlegrounds. Before we get into the nitty-gritty of resistance, let's set the scene. Where do we find this troublesome bacterium? The short answer is everywhere, but its impact varies dramatically. In the cattle industry, certain pathogenic strains of E. coli are a primary cause of calf diarrhea, a devastating disease that can lead to high mortality rates and significant economic losses for farmers. This isn't just a problem for newborn calves. Studies have shown that cattle can be important reservoirs for antimicrobial resistant bacteria, which can then pose a risk to human health through the food chain. The story is similar in poultry production. E. coli infections like colobacillosis can cause respiratory and intestinal diseases, leading to septicemia and major financial costs for the broiler industry worldwide. And in pig farming, colistin, an antibiotic of last resort in human medicine, has been historically used to control E. coli infections like post-weaning diarrhea. This widespread use, as we'll see later, has had serious consequences. But it's not just farm animals. Our closest companions, our dogs and cats, are also part of this story. Given the close physical and emotional bond we share with our pets, they can serve as a potential bridge for resistant bacteria to enter our homes and our lives. A major study in Japan, for instance, investigated third-generation cephalosporin-resistant E. coli from diseased dogs and cats, finding alarming connections to strains that infect humans. Perhaps most surprisingly, this battle is also being fought in the wild. You might think that wildlife, far from human intervention and antibiotic use, would be a pristine reservoir of susceptible bacteria. But research tells a different story. A comprehensive study in Poland sampled E. coli from wild boar, deer, and even European bison. While the levels of resistance were generally lower than in farm animals, the researchers still found bacteria armed with sophisticated resistance genes for critical antibiotics like cephalosporins. This suggests that our impact, the selective pressure from antibiotics used in agriculture and human medicine, is bleeding out into the natural environment, creating what scientists call a resistome, a vast shared pool of resistance genes. And finally, even the processing of animal waste in rendering plants has been identified as a significant source and potential spreading point for these hardy, resistant microbes, including dangerous clones that are known to cause severe infections in humans. So, from the farm to the family pet, and from the forest to the factory, E. coli is present, and where it is present, it is learning to resist. But how exactly does it do it? Segment 2. An arsenal of resistance, the molecular machinery of a superbug. To understand how to fight antimicrobial resistance, we first need to understand the enemy's weapons. And E. coli has a truly formidable arsenal, developed and refined through decades of evolutionary pressure. Let's break down its key strategies for defeating some of our most important classes of antibiotics. Fighting the beta-lactams, breaking the ring. First up, we have the beta-lactams. This is a huge and critically important family of antibiotics that includes penicillins and cephalosporins. You've almost certainly taken a beta-lactam antibiotic at some point in your life. They work by targeting the construction of the bacterial cell wall, causing it to weaken and burst. But E. coli has a devastatingly effective countermeasure, enzymes called beta-lactamases. These are molecular scissors that chop up the beta-lactam antibiotic, rendering it useless. Over the years, E. coli has developed increasingly powerful versions of these enzymes. One of the most worrying groups is the extended-spectrum beta-lactamases, or ESBLs. These enzymes can destroy not only basic penicillins, but also more advanced, third-generation cephalosporins, drugs we rely on to treat serious infections. Studies from around the world have documented the alarming spread of ESBL-producing E. coli. In Poland, researchers found them in healthy food animals like broilers, turkeys, and pigs. In Thailand, ESBLs were highly prevalent in both sick and healthy pigs, with nearly half of the isolates from sick pigs producing these enzymes. And it's not just livestock. ESBLs have been found in wildlife and are a growing concern in companion animals. The genes that code for these enzymes have names like BLA-CTXM, BLA-SHV, and BLA-TM. The BLA-CTXM family, in particular, has become the dominant type of ESBL worldwide. For example, a Polish study on food animals found BLA-CTXM1 to be one of the most common resistance genes, while a study on wild animals in the same country found both BLA-CTX1 and BLA-CTX15. But the story doesn't end there. E. coli has another trick up its sleeve: AMP-C beta-lactamases. These enzymes are resistant to the inhibitors we often pair with beta-lactam antibiotics, making infections harder to treat. The gene BLA-CMY2 is a particularly notorious plasmid-borne AMP-C gene. It has been found in E. coli from poultry, pigs, and cattle across Europe, and has even been detected in wild boars and deer in Poland. A study conducted at an animal rendering plant also identified E. coli strains carrying the BLA-CMY2 gene, highlighting how waste processing facilities can become hotspots for the dissemination of these resistance mechanisms. Dodging the quinolones, target alteration, and pumping drugs out. Next, let's turn to another class of powerful, broad-spectrum antibiotics, the quinolones and their more advanced cousins, the fluoroquinolones. These are entirely synthetic drugs, and for a long time, they were our go-to weapons against many gram-negative bacteria, including E. coli. They work by targeting two essential bacterial enzymes, DNA gyrase and topoisomerase 4, which are responsible for managing the complex coiling and uncoiling of DNA during replication. The quinolones essentially jam this machinery, leading to lethal breaks in the bacterial DNA. So how does E. coli get around this? Its primary strategy is to change the lock so the key no longer fits. It develops mutations in the genes that code for these enzymes, specifically in regions called the quinolone resistance determining regions, or QRDRs. The key genes here are GIRA and PARC. A single mutation in GIRA can give the bacterium a low level of resistance. But the real danger comes with accumulation. A second mutation in GIRA, often followed by mutations in PARC and another gene called PAIR, can lead to high-level resistance, making the drugs completely ineffective. A major study of E. coli from companion animals, pet owners, and non-pet owners in Korea found that this stepwise accumulation of mutations was the single most important factor driving high-level resistance. In fact, the number of mutations was strongly correlated with how resistant the bacteria became. But that's not its only defense. E. coli can also deploy efflux pumps. Imagine these as tiny, molecular sump pumps embedded in the bacterial cell wall. Their job is to actively pump out toxic substances, including antibiotics, before they can reach their target. The aceorbital efflux system is a major player here, capable of pumping out a wide range of drugs. Some studies have found that overexpression of these pumps contributes to quinolone resistance, though other research suggests that once high-level resistance is achieved through target mutations, the pumps may play a less significant role. There's also a third, albeit less common, mechanism: plasmid-mediated quinolone resistance, or PMQR. This involves resistance genes, like the QNR family, that are carried on mobile pieces of DNA called plasmids. While these genes have been detected in E. coli from both wildlife and companion animals, several studies have found their prevalence to be quite low, suggesting that for quinolones, target site mutation remains the king of resistance mechanisms. Resisting the last resort, the rise of colistin resistance. Now we come to one of the most concerning stories in modern medicine: resistance to colistin. Colistin is an old antibiotic, part of the polymyxin family, that was largely shelved for decades due to its potential toxicity to the kidneys. However, with the rise of multi-drug resistant bacteria that could shrug off nearly everything else, doctors were forced to bring it back as a last resort treatment for life-threatening infections. For years, resistance to colistin was rare and thought to be caused only by slow chromosomal mutations. Then, in 2015, everything changed. Researchers in China discovered a gene called MCR1, which conferred resistance to colistin and was located on a plasmid. This was a watershed moment. A resistance gene for a last resort antibiotic that could be easily transferred between bacteria was a public health nightmare. Since that discovery, the story has only grown more complex. We now know of multiple MCR genes, from MCR1 all the way up to MCR10. The sources you've provided paint a vivid picture of their spread through animal populations. A large study of E. coli from pigs in Thailand found an astonishingly high prevalence of MCR genes. The most common was not MCR1, but MCR3, which was present in 45% of all isolates. This suggests that in some regions, other MCR variants may be more important than the original MCR1. The study also found MCR1 and, for the first time in 2007, MCR2. Even more worrying, they found isolates carrying multiple MCR genes simultaneously, such as MCR1 and MCR3 together. This isn't limited to pigs. A study in Turkey identified the MCR4 gene in E. coli from cattle, marking the first report of this specific gene in cattle in that country. The global picture shows that while MCR1 is the most widespread variant, others like MCR2 and MCR4 have been found mainly in Europe, while MCR3 seems more common in Asia. This geographical diversity highlights how these genes are evolving and spreading differently around the world, making surveillance a huge challenge. A surprising twist: how losing a gene can increase susceptibility. Most of the time, resistance is about gaining something, a new gene, a mutation, an overactive pump. But what if a bacterium could become more susceptible to an antibiotic by losing a gene? That's exactly what a fascinating recent study discovered about E. coli's relationship with another important class of antibiotics, the aminoglycosides, which include drugs like gentamicin and amikacin. The gene in question is HNS, which codes for a protein called HNS. HNS is a global regulator, a master switch that controls the expression of many other genes, often silencing foreign genes that the bacterium has acquired. Researchers created a mutant strain of E. coli with the HNS gene deleted and were stunned by the result. The mutant bacterium, strain E1HNS, became incredibly sensitive to aminoglycosides. Its minimum inhibitory concentration, or MIC, dropped by 8 to 16 times. It was as if removing a key piece of its genetic architecture had left it vulnerable. So what was happening? The investigation revealed a multi-pronged breakdown in the bacterium's defenses. First, without HNS, the bacterium's outer membrane became more permeable. Scanning electron microscope images showed that the mutant cells were deformed and damaged, creating more entry points for the antibiotic to get in. Furthermore, the expression of genes for porins, protein channels in the outer membrane, like OMP-C, OMP-F, and OMP-N went up, effectively opening the floodgates for the drug. Second, the activity of the efflux pumps, which we discussed earlier, was significantly inhibited. Genes for several efflux pumps, including ACRA, ACRB, and MRE, were downregulated. So, not only was more drug getting in, but less of it was being pumped out. And third, and perhaps most elegantly, the bacterium's own energy metabolism turned against it. Aminoglycosides need energy to cross the inner bacterial membrane, and this energy is supplied by what's called the proton motive force, or PMF. In the HNS-deleted mutant, key metabolic pathways like glycolysis and the electron transport chain were kicked into high gear. This generated a much higher PMF, which then actively pulled more and more aminoglycoside molecules into the cell, leading to its demise. It's a beautiful example of how disrupting a single, central regulatory system can cause a cascade of effects that completely change a bacterium's relationship with an antibiotic. This finding not only deepens our understanding of resistance, but also opens up a potential therapeutic avenue. If we could find a drug that inhibits the HNS protein, we might be able to resensitize resistant bacteria to aminoglycosides. Segment 3. The social network of bacteria, biofilms, and quorum sensing. We often think of bacteria as individual, free-floating cells, but in reality, they are highly social organisms. They can form complex, structured communities called biofilms, and they can communicate with each other using a chemical language in a process known as quorum sensing. Both of these social behaviors have profound implications for antimicrobial resistance. Let's start with biofilms. A biofilm is essentially a city of bacteria. The cells attach to a surface and begin to secrete a slimy, protective matrix made of sugars, proteins, and DNA. This matrix encases the community, acting like a fortress. This structure is a major headache in medicine because it makes bacteria incredibly resistant to antibiotics. The physical barrier of the matrix can prevent drugs from even reaching the cells deep inside. Furthermore, bacteria within a biofilm often have a slower metabolism, which makes them less susceptible to antibiotics that target active cellular processes. A study focusing on E. coli isolated from calves with diarrhea found a significant link between the ability to form a biofilm and multi-drug resistance. Over 94% of the isolates that were strong biofilm formers were also resistant to multiple classes of antibiotics. This highlights how biofilm formation can be a key survival strategy for resistant pathogens. But the same study also offered a glimmer of hope. They investigated an antibiotic called Cefoxitin, a type of cephalosporin. They discovered that Cefoxitin, at concentrations that didn't kill the bacteria, could effectively inhibit the formation of biofilms. By observing the bacteria with a powerful laser scanning confocal microscope, they saw that the drug disrupted the initial adhesion of the bacteria to surfaces, preventing the biofilm city from ever being built. This suggests a potential strategy, using certain antibiotics not just to kill bacteria, but to disrupt their community structures and make them more vulnerable. The mechanism behind this effect appears to be linked to the second social behavior we're going to discuss, quorum sensing. Quorum sensing is how bacteria talk to each other. They release small signaling molecules called autoinducers into their environment. As the bacterial population grows, the concentration of these molecules increases. Once it reaches a certain threshold, quorum, it triggers a coordinated change in gene expression across the entire population. It's a way for them to act in unison, to launch a virulent attack, or to form a biofilm, for instance. In gram-negative bacteria like E. coli, one of the most studied quorum sensing systems uses signaling molecules called acylhomoserine lactones, or AHLs. The study on cefoxitin found that the drug inhibited biofilm formation by downregulating the expression of key genes involved in quorum sensing, like LuxS, as well as genes directly responsible for building the biofilm components, such as CSGD, which controls the production of curly fibers that help bacteria stick together. But here's where it gets even more interesting. Quorum sensing doesn't just help bacteria build defenses, it can actively increase their resistance. Another study looked at E. coli causing calf diarrhea and investigated the effect of adding an AHL signaling molecule, specifically one called C8, to the bacteria's environment. They found that when they added C8, the bacteria's resistance to the fluoroquinolone antibiotic ofloxacin increased dramatically. The minimum concentration of the drug needed to kill the bacteria shot up from 32 micrograms per milliliter to 128. By analyzing the bacteria's entire set of expressed genes, its transcriptome, the researchers pieced together how this happens. They believe the AHL signaling molecule triggers two key responses in the bacteria. First, it appears to activate the SOS response, which is the bacterium's emergency DNA repair system. Since ofloxacin works by causing DNA damage, ramping up the repair mechanisms helps the bacteria survive the assault. Second, the signaling molecule seems to influence another regulatory network called the CPXAR2 component system, which is known to control biofilm formation and the expression of multi-drug efflux pumps. Essentially, by listening to the quorum sensing signals, the E. coli cells were able to preemptively bolster their defenses, repair damage more effectively, and pump out the drug, all leading to enhanced resistance. These findings open up a whole new frontier in our fight against resistance. If we can develop drugs that jam these communication signals, quorum quenchers, we might be able to disarm bacteria, prevent them from forming biofilms, and make them susceptible to antibiotics once again. It's a strategy that targets their social network rather than trying to kill them directly, and it could be a powerful new tool in our arsenal. Segment 4. The Resistance Reservoir, a One Health Perspective. We've explored the intricate molecular machinery that E. coli uses to defy our antibiotics. But where does all this resistance originate and fester? The answer lies at the heart of the One Health concept, connecting livestock, pets, wildlife, and humans in a complex web of transmission. Let's take a tour of these resistance reservoirs. On the farm, food animals as a crucible for resistance. It's no secret that antibiotics have been used extensively in food animal production, not just for treating sick animals, but also for preventing disease in entire flocks or herds, and, until recently in many places, for promoting faster growth. This widespread use creates an intense selective pressure, a perfect environment for resistant bacteria to thrive and multiply. The evidence for this is overwhelming. Studies from major poultry-producing regions like the US, Brazil, China, and the EU consistently show high rates of resistance in E. coli isolated for broilers. A global overview revealed that resistance to older antibiotic classes like tetracyclines, sulfonamides, and penicillins is extremely common, often exceeding 40% in most countries. A study in Poland found that over 70% of E. coli from broilers were resistant to ampicillin and the fluoroquinolone ciprofloxacin. In China, resistance rates to tetracycline and ampicillin in broiler isolates often soar above 80%. Pigs are another major reservoir. The Thai study we discussed earlier found that 97.5% of E. coli from healthy pigs were multi-drug resistant. The extensive use of colistin in piglets to prevent diarrhea has been directly linked to the high prevalence of MCR genes, with a staggering 95% of piglet isolates showing resistance to this last resort drug. The story in cattle is similar. A study in Turkey collected fecal samples from cattle on 12 different farms and found widespread resistance. Every single E. coli isolate was resistant to the cephalosporin cefotaxime, and over 95% were resistant to ampicillin. The researchers also identified the MCR4 colistin resistance gene, a first for cattle in that country, highlighting how these genes are continuing to spread into new animal populations. The link between antibiotic use and resistance levels is not just theoretical. We have concrete evidence. In France, where detailed data on antibiotic sales for poultry is available, a clear trend emerged. As the use of tetracyclines decreased between 2006 and 2016, the rate of tetracycline resistance in E. coli from broilers also fell significantly. Conversely, in the United States, the use of fluoroquinolones in poultry was banned in 2005. The result? Resistance rates to these drugs in U.S. broiler isolates are now below 5%, a stark contrast to the over 40% resistance seen in Brazil, China, and the EU, where their use is permitted. These examples provide powerful proof that reducing antibiotic use in agriculture can have a direct and positive impact on reducing resistance. In our homes, the role of companion animals. Moving from the farm to the family room. Our pets are also caught up in this cycle. Because we share our homes, our beds, and our lives with them, dogs and cats can act as vectors, potentially transmitting resistant bacteria to their human companions. A major Japanese study provides a compelling case. They analyzed cephalosporin-resistant E. coli from sick dogs and cats and made some striking discoveries. The most common resistance genes they found were BLAST-CTX, M27, BLAST-CTX, M14, and BLAST-CTX, M15. This is significant because these are the same genes that are predominant in human infections in Japan. Even more telling, they identified the pandemic clone ST-131, a globally successful and often highly resistant lineage of E. coli, in over a third of the pet isolates. Genetic analysis showed that an ST-131 strain from human in Japan was very closely related to the cluster of strains found in these pets, suggesting a shared reservoir and potential for cross-species transmission. Another study, this time from Korea, looked at quinolone resistance in pets and their owners. They found that the molecular mechanisms of resistance, specifically the pattern of mutations in the gyra and PARC genes, were identical in the isolates from companion animals and humans. This doesn't mean that pets are always the source. The transmission can go both ways. But it underscores that we are dealing with the same pool of resistant bacteria. Therefore, the prudent use of antibiotics in veterinary clinics is just as critical as it is in human hospitals and on farms. In the wild, an unseen reservoir. Finally, let's venture into the wild. As we touched on earlier, the natural environment is not immune to the problem of resistance. The Polish study of wild boar, deer, and bison provides a fascinating window into this phenomenon. They found E. coli carrying genes for ESBLs like BLAST-CTX, M1, and BLAST-CTX, M15, AMC enzymes like BLAST-CMY2, and even plasmid-mediated quinolone resistance genes like QNRS. How did these sophisticated resistance mechanisms end up in animals that are rarely, if ever, treated with antibiotics? The researchers concluded that it's not from direct transmission from farm to forest. Instead, they point to a more insidious cause: environmental contamination. Manure from treated livestock used as fertilizer, runoff from farms, and wastewater from human settlements all carry a load of antibiotics and resistant bacteria into soil and water. This creates a low-level, persistent selective pressure in the environment, favoring the survival and spread of resistant microbes. Wild animals, through their foraging and drinking habits, then pick up these resistant bacteria. For instance, the study noted that wild boar and roe deer, which often feed on agricultural lands bordering forests, had higher rates of resistance than red deer and bison living deeper in the forests. This shows that wildlife is not just a passive victim, but an active part of the cycle. They can act as a melting pot, where resistance genes from different sources can mix, and as a vector for disseminating these genes across vast geographical areas. This interconnectedness, from the piglet in a farrowing crate to the wild boar in the forest to the dog on our lap, is the essence of the One Health Challenge. The resistance problem can't be solved by focusing on just one area. It requires a coordinated effort across human medicine, veterinary medicine, and environmental stewardship. Segment 5. The superhighways of spread, plasmids, clones, and the global picture. We've seen how E. coli resists and where this resistance thrives. Now let's look at how it spreads. Resistance wouldn't be such a global crisis if every resistant bacterium had to invent its defenses from scratch. The real danger lies in its ability to share its genetic weapons. The primary vehicles for this are plasmids. Plasmids are small, circular pieces of DNA that exist separately from the bacterial chromosome. They are mobile genetic elements, capable of being copied and transferred from one bacterium to another, even across different species. A single plasmid can carry genes for resistance to multiple classes of antibiotics, turning a susceptible bacterium into a multi-drug-resistant superbug in a single transaction. The sources we've examined are filled with examples of these genetic superhighways. In the study of Polish food animals, researchers found that cephalosporin-resistant genes like BLASH12 and BLAST-CTX1 were located on highly transferable INCX1 plasmids. The ease with which these plasmids could be transferred led the authors to suggest that the spread of resistance might be better understood as epidemics of certain plasmids, rather than just the spread of resistant bacterial clones. Similarly, the study of Japanese pets found that many of the resistance genes, including BLAST-CTX27, were located on INC-F plasmids, which also carried a cocktail of genes for resistance to aminoglycosides, sulfonamides, and tetracyclines. In wildlife, INC1, alpha plasmids were found carrying AMP-C resistance genes. This constant shuffling and sharing of plasmids creates a dynamic and ever-evolving landscape of resistance. While plasmids are the vehicles, certain bacterial strains have become exceptionally good drivers. These are often referred to as high-risk or pandemic clones, lineages of E. coli that have been remarkably successful at spreading across the globe, accumulating resistance genes as they go. The undisputed king of these is sequence type 131, or ST131. This clone is a major cause of extraintestinal infections in humans worldwide, and it is notorious for its multi-drug resistance, particularly its frequent association with the ESBL gene BLAST-CTX-M15. Our sources show that ST131 is not confined to humans. It was a dominant clone found in sick dogs and cats in Japan, where it was often carrying BLAST-CTX-M27. Perhaps even more alarmingly, the study of an animal rendering plant also identified CMY2, producing E. coli belonging to the ST131 lineage. Finding this pandemic human pathogen in an industrial environment that processes animal waste is a stark reminder of the zoonotic potential and the serious public health risk these clones represent. Another clone mentioned is ST648, which has also been described as a pandemic clone in dogs and cats, known for its ability to combine multi-drug resistance with virulence. Finally, this global spread has led to fascinating geographical variations in resistance patterns. For instance, in Europe, the most common CEXM-type ESBLs in companion animals are BLAST-CTX M1 and BLAST-CTX M15, while in Asia, BLAST-CTX M14 and BLAST-CTX M15 are more prevalent. This highlights how different resistance genes can become dominant in different ecological and geographical settings, likely influenced by local antibiotic prescribing habits in both human and veterinary medicine. Interestingly, the link between animal and human resistance isn't always a straight line. The Polish study on food animals made a crucial observation. While genes like BLASTCTX1 were common in the animals they tested, the dominant gene in human clinical infections in Poland for decades has been BLASTCTX3. This discrepancy suggests that while animals are a massive reservoir of resistance genes, the direct transfer of specific resistant strains into the human clinical setting may be complex and not always the primary driver of human infections in every case. Instead, it points to a broader ecological problem, where the entire environmental resistem is enriched with resistance genes, which can then be picked up by pathogens in both animals and humans. Conclusion: a call for unified action. We've covered a lot of ground today, from the microscopic world of DNA mutations and efflux pumps to the global landscape of livestock production and wildlife ecosystems. The picture that emerges is one of breathtaking complexity, but the central message is starkly clear. Antimicrobial resistance in Escherichia coli is a formidable and multifaceted challenge that transcends species and borders. We've learned that E. coli employs an incredible diversity of strategies to survive our antibiotic onslaught. It produces enzymes like ESBLs and AMPCS to dismantle our most trusted cephalosporins. It alters the very targets of quinolones within its own DNA, building resistance one mutation at a time. It has even acquired mobile genes to defeat our last resort antibiotic, colistin. We've seen how it can manipulate its own internal systems, like the HNS protein, to control its susceptibility, and how it uses social behaviors like biofilm formation and quorum sensing to build resilient communities and coordinate its defenses. We've also seen that the reservoirs for this resistance are all around us. The heavy use of antibiotics in food animal production has created a powerful engine for the selection and amplification of resistant strains, as evidenced by studies from every major agricultural region in the world. Our companion animals, living in our homes, share a pool of resistance genes with us, with pandemic clones like ST131 moving between pets and people. And our impact has reached deep into the natural world, contaminating the environment and turning wildlife into an unforeseen reservoir and vector for resistance genes. The rapid spread of these defenses is fueled by the superhighways of horizontal gene transfer, plasmids that carry resistance cassettes from one bacterium to another, creating novel combinations of resistance and spreading them far and wide. So what can be done? The sources point not to despair, but to a clear path forward, built on the principles of One Health. The first and most critical step is the prudent and responsible use of antibiotics in all sectors: human, animal, and agricultural. The examples from France and the US show us that reducing antibiotic use can lead to a tangible decrease in resistance levels. The experience in Denmark, where the removal of antibiotic growth promoters was followed by a drop in quinolone resistance, is another powerful testament to this principle. We also need robust, harmonized global surveillance. The inconsistencies in how resistance is monitored from one country to another make it difficult to get a clear global picture. Standardized monitoring in animals, humans, and the environment is essential to track emerging threats and measure the impact of our interventions. And finally, we must continue to innovate. Research into novel strategies, like drugs that inhibit biofilm formation or block bacterial communication through quorum quenching, offers new hope for outsmarting these ancient survivors. The discovery that disrupting the HNS protein can resensitize E. coli to aminoglycosides is a perfect example of how a deeper understanding of bacterial biology can reveal new vulnerabilities to exploit. The challenge is immense, but it is not insurmountable. By recognizing the intricate connections between our health, the health of our animals, and the health of our planet, we can begin to build a unified strategy to preserve the miracle of antibiotics for generations to come. Thank you for listening. The titles for the reference papers are in the video description. From VetMed Digest, where we keep minds inspired, hearts light, and tails wagging,